Thank you. Well, that was a quick coffee break. Welcome back, everyone. I decided not to talk about the importance of studying long-lived animals today, even though I just wrote a book on it because, first of all, Vera Gorbanova was going to do that after me. I thought that I would really talk about something that I think is very important at this stage in aging research. This is the most exciting time in the aging field that I can remember by a long shot. We have lots and lots and lots of leads to things that keep people potentially healthy longer. But what I'd like to do today is I'd like to manage expectations a little bit, because I think it's important, because it's been a long, hard fight to earn academic respectability in this field. And I think we've gotten there, and I don't want to see us squander it. So I'm going to talk about why human trials based on animal studies so often fail. And John Speakman made this point to some extent. And this is one of the consequences of failures. This is the stock price of a company that had a very optimistic expectation of a drug they had, and it didn't pan out, and the stock price went down like crazy. So the historical rate of failure of clinical trials that are based on animal studies is extraordinarily high. For cancer, several reviews have come up that about 92% of all clinical trials based on, we're very, based on mouse studies, we're very good at curing cancer in mice. We're pretty good people. For stroke, the failure rate has been over 99%. For Alzheimer's disease, also over 99%. Now, in any other field, in business, for instance, this kind of failure rate would make people rethink the basic research paradigm that they're using. But that hasn't happened in biomedicine. And I'd like to examine a few of the reasons why I think there's such a high failure rate and what we can do to potentially manage it. Geroscience, we don't know. Maybe it won't be so high for geroscience. Maybe because we use all these model species besides mice, maybe it will be better. All of them age and we age, maybe it'll be better. But we don't know that because we don't yet have a single human trial that's shown success at enhancing and extending human health. There's two issues, I think. One is a species. Basically, we've collapsed the entire 6,000 species of mammals down to one experimental species, the mouse. In the case of the C57 Black 6, I would call it, with my friend Rich Miller, a mouse-like object. But the thing about the mouse that's relevant to this is that it's one of the least successful, most cancer-prone mammals on the planet. And yet we're using it in the hopes that if we can make it a little bit more successful, that will lead to something that allows us to be a little bit more successful at aging, even though we're the longest lived terrestrial mammal already. We're already very good at aging. We want to get better at resisting aging, but we're already pretty good. So I'm not going to talk about the species issue. I don't think there's anything specific about the mouse except we only use one species. I think that's the key thing. What I really want to talk about, though, is the environment, because that's something that we tend to ignore in biomedical research. We tend to treat animals like they're books or bookends, that you could throw them in anywhere and they'll be just as happy. And that's not the case. It's not the case at all. And I'm going to show you some reasons. First of all, why we use these kinds of environments. There's very good scientific rationale for why we use these kind of environments, but it's also antithetical to translation, to actually using the results to examine the possibilities of human health benefits. So there is a reproducibility crisis, not just in our field, other fields, cancer, psychology. I think we don't know how bad this is in our field because we don't have a great culture of replication in our field. And so one of the things that when people say, well, we have 20 or 30 or 40 drugs that slow aging, and I always go, wait a second. I don't believe that, because to me, something has to be replicated in at least a couple of independent labs and probably in a couple of genotypes before I'm ready to believe it. So this is a real problem. Now, how do you deal with the problem? One of the ways you deal with the problem is what the interventions testing program has done, which is you standardize the environment to an incredible degree. And they've done that. And they do the same experiment in 3 places, so they have instant replication. And they're very, very good. They're very, very good at this. If you look at these survival curves, if you've ever done survival experiments yourself, getting this kind of consistency in 3 independent experiments, even if it was in the same lab, would be remarkable. They've managed to do it at these 3 distinct places. So that's very, very good, and it's led to, in this case, a high degree of replicability. But it comes at a cost. That kind of standardization is good for replication. It's good for investigating mechanisms, but it's bad for translation. Because what we've got is we've got a laboratory environment that's absolutely unchanging. It's a little bit chilly for the mice. They're confined to a degree that would be prisoners in solitary confinement have more of an opportunity for physical activity. They're microbiologically depauperate. I'll talk about the microbiome in a while. They're unstimulated by any sensory input except one another. And this is very, very different from human environment, but it's also very, very different from a mouse's normal environment. And I think that has consequences. Because for humans, we live in different environments, we have different health habits, we have different diseases we've been exposed to, different genetics, all kinds of variability. Now, human clinical trialists make an important distinction between several kinds of intervention trials. Explanatory trials are those to determine whether an intervention has an effect under very specific, narrow conditions. Most phase 1 trials are like that. You get one group of people in one facility and you treat them exactly the same. So you highly select the subjects. It's very, very good for replication. But the thing is, it may not be generalizable. The other type of study, and the kind that's typically used in phase 3 trials, is what's called a pragmatic trial. A pragmatic trial is measure how well an intervention works under real-world conditions, and I think that's what we're looking for. We're not looking for an intervention that only helps, college professors and insurance executives. We'd like to find interventions that extend the health of everybody. There's broad participants, clinically relevant outcomes, and a tremendous variation in the environment, both the internal and external environment in which the trials take place. So virtually all preclinical trials are explanatory trials. And sometimes, by accident, you find one that isn't. And when it isn't, there's remarkable results. So this is a study of Huntington's disease mouse. Now, Huntington's disease is considered to be the classic genetic disease. If you have a Huntington with more than a certain number of glutamine repeats, you're going to get the disease, period. And here's a successful intervention. The red line there shows the age of onset and the percent of onset of control animals. And as you can see, this intervention has cut down the age of onset and the rate of onset dramatically. So what is that intervention? It's simply putting a few tubes and toys in a cage. That amount of stimulation, of cognitive and sensory stimulation, has a huge effect. Now, if you talk to neuroscientists, they know all about this. Most of the Alzheimer's models, the mouse Alzheimer's models, if you put them in an enriched environment like this, the phenotype goes away. So I've asked a number of them, well, if everybody knows this in the field, why doesn't everybody enrich their environments? And they say, well, it makes the effect that we're looking for trivial compared to the effect of the enrichment, which I don't find a very satisfying answer. I don't know about you. And here's another aspect, and this really boggled my mind, as I hope it will boggle your minds if you haven't heard me speak about it before. A recent study of senolytics in the effect of COVID-19 in mice found the following, that if you took the mice that were standard black 6 mice but were old mice, and you simply put them on bedding from pet store mice, perfectly healthy pet store mice, within 2 weeks they were all dead. Think about that. Perfectly healthy pet store mice, you put these mice on their bedding and they're all dead in 2 weeks. And this brings me to really a provocative case of failed translation, and one that's been in the news recently. 2006, there was a study done. This monoclonal antibody to be used for treatment for leukemia and rheumatoid arthritis was given in a phase 1 trial. Now this had flown through all the animal work. 6 men were infused with it, and within 1 hour they all collapsed of massive cytokine storms. Multiple organ failure. Fortunately, none of them died. A couple of them were in the hospital for a long time. But what happened? Because this had flown through the animal trials. Well, it turned out that a bit later, somebody took the same mice and simply exposed them to wild mice, gave them the microbiome of wild mice, and they got exactly the same cytokine storm that the humans had. So this is a dramatic example of the fact, and this is just how they gave them the microbiome. They basically put black 6 animals in wild mothers, but there are much simpler ways to do it. What this means is that in our desire to make specific pathogen-free mice, which have been critical to replication, to successful replication in a variety of fields, in protecting the mice from pathogens, we're also protecting them from a normal microbiome. And what you're looking at here is simply differentiated CD8 T cells in laboratory mice compared to neonatal humans and adult humans. So basically what we have given is we've given adult mice a neonatal immune system because they haven't had the exposure to even the commensal microbes. It's easy to fix this. We simply put them in with a pet store mouse. We co-house them with a pet store mouse, and suddenly they have an adult immune system. This is another case where they did that, and even these are young mice. Simply putting them, co-housing them with pet store mice, about 20% of them die, just because they have no immune system, so to speak. So it's easy for us to fix this. We simply need to understand that they don't have a normal microbiome, that having a normal microbiome not only is important for resisting infectious diseases, it's important for lots of stuff. It's important for, basically, for the microbiome itself. There's a yin and yang, a give and take between the microbiome and all kinds of things in the immune system. Sterile inflammation, cancer resistance, all of these things are a function of having a normal, or at least a quasi-normal microbiome. So I'd like to point that just this one environmental change makes a tremendous difference, and it's likely to make a tremendous difference in aging as well. Now, I focused on that because I think these are pretty dramatic and vivid examples of the effect that small changes in the environment can have on our experimental research. But I'd like to point out that it's not just that we don't give animals really normal circadian rhythms. We flick the light off at a certain time, we flick it on. We don't let the temperature vary. We don't let it happen gradually. We don't have seasonal differences. And we know that seasonal circadian differences affect all of these things that I've listed there. We also keep them at a constant temperature. I don't think it's the temperature itself that's the problem. I think it's the variety of temperatures that's important. And of course, if we give them an exercise wheel, that changes everything. So let me just finish up here by saying a couple of things. What can be done about it? First of all, I think we need to test all of our interventions that we think are going to work in multiple genotypes. Multiple genotypes in mice. I actually had multiple species on here as well, because I think multiple species is important, but that's the topic of a whole other talk. I think we need to use more realistic environments. You don't necessarily need to put lab mice in with pet store mice, but you can put them in, even if you just put them in dirt, they get a normal microbiome. And multiple environments, enriched environments. Here's a couple of examples of an enriched environment. I think this is critically important. Now, if you do this, are they all going to live on average 900 days? No, some of them are going to be short lived, some of them are going to be long lived. You know, I'd like to basically challenge what Brian Kennedy said earlier, which is, well, it's only these things that if they don't live to be 900 days, it's not really an interesting result. Well, I think what we want to do is we want to develop interventions that are good for us privileged, long-lived people, but also for disadvantaged people. You know, if we had only focused on the healthier people, where would we be with antihypertensives? Where would we be with insulin? Where would we be with another thing? So I think we really need to develop translational intervention paradigms for animals. And if we do that, I think we can lower that frequency of failed translational experiments substantially. So thanks, I'll stop there. Here's the book that I didn't talk about. Soon to be out in Chinese, by the way. I thought I would mention that. If you haven't read it in English, please try to get it in Chinese. Thanks a lot for your attention.
So I'm convinced that the wild animals will be better tools. But what's the blockage for people adopting that, other than need to consider many things, like you have to give them drugs for infection, have the colony expanded. But if you have maybe not a perfect SPF environment, would that be okay? So why we need to control the bacteria? We just worry about the virus, it may be okay.
Yeah, I mean, clearly you don't want to get some pathogenic virus in there that kills everything.
But you can provide them with the bacteria.
But they're commensal viruses as well. So I don't think there's any barrier to stop. Well, there is a barrier. The barrier is study sections. People are used to doing things a certain way, and they don't like it when you talk about changing it. And this is a paradigm that's been around for a long, long, long time, and it was developed for mechanistic studies. And when we started to think about translation, we never thought about altering the paradigm. We just used the same old paradigm. And I think it's just because it's easiest to continue to do things the way you've always done them, rather than to think about, well, is there a better way? Like I said, if this were business and you had a 95% failure rate, they'd say, okay, we got to do something different. For some reason in the biomedical community, that hasn't happened, but I'm hoping it will.
All right.
I have a question.
Was it reason?
Sorry, I have a question, Steve. Thank you so much for this wonderful talk. So the question I have is, this complicates effectively already complicated longevity drug discovery ecosystem. Now, for a company that lives on shoestring budget and still wants to discover longevity therapeutics, how can some of these principles that you talked about be extended with practical intentions in mind?
Yeah, I mean, that is a good question, and it certainly will make preclinical trials a little bit more expensive if you figure that they should be done in multiple environments. But think about the cost of doing this opposed to a phase 3 human trial that fails. It's a drop in the bucket. And if you could lower the failure rate to even 50%, then I think ultimately it would be a money saver. Although at the level of a single company, I don't know if that's any consolation.
The last question goes to Professor Speakman.
Yeah, great talk. Could I just ask what your feeling is about social housing? There's been social housing, so housing rodents together. So, yes. I mean, as a sort of anthropomorphism, it seems like we shouldn't keep animals on their own forever through the whole life. But on the other hand, male mice in the wild never come within a meter of each other without killing each other. And so sticking them together in a little cage together is probably not very natural. I just wonder what your feelings are about that whole thing.
Well, first of all, I think it's a real issue. I think social housing is important. Now, the way you do it will depend on the mice you're using, the facilities that you have. I think getting rid of the shoebox cage is a big step in the right direction. One thing I didn't mention is that all of our experimental mice have never reproduced. And that's another consideration that makes them very, very different. So I just think we need to rethink this from the standpoint of what's biologically important. Because I think if the more biologically realistic we can make it for mice, the more likely it's going to translate to us.
Many thanks goes to Professor Steven Austad for